So we've heard a lot about DNA methylation profiles, and my lab is actually interested in studying the effectors of epigenetic modification. So my lab is interested in using a quantitative mass spec as a technology to identify chromatin readers. And chromatin readers, we've heard about them already in this meeting. Chromatin readers are defined as proteins that carry domains that can specifically recognize epigenetic modifications. So these can be histone modifications or also proteins recognizing methylated and hydroxymethylated DNA. So we use this technology to identify these readers, and we also use the same technology to characterize the protein complexes that these readers form, because it turns out that these chromatin readers are not doing their job alone, but often assemble into large multi-protein complexes. We study the function of these chromatin readers in differentiating stem cells and also in relation to cancer, because it becomes apparent that epigenetic associated proteins and also these chromatin readers are often deregulated in cancer. So today's talk will focus on our efforts to identify chromatin readers for methylated and hydroxymethylated DNA. So this picture is sort of a textbook model of how we think DNA methylation regulates transcription. So we have methylation mostly occurring on CG dinucleotides, which are concentrated on so-called CpG islands, which are often in front of promoters. And what we know is that when these CpG islands are methylated, that the gene that is upstream from the CpG island is transcriptionally silenced, and if the CpG island is unmethylated, then the gene is active. And we think that this is due to the fact that these methylated CpG islands can recruit specific proteins, so readers for these methylated CpG islands that upon recruitment set up a transcriptionally silent chromatin configuration, and this then results in transcriptional silencing. And non-methylated CpGs can also recruit specific proteins. Often these proteins carry a so-called CXXC motif, and these proteins upon recruitment establish an active chromatin configuration, including K4 trimethylation and high levels of histone acetylation, and this then results in transcriptional activation. Now, a couple of years ago, the story became a little bit more complicated because the TET enzymes were discovered, and they catalyzed the enzymatic conversion of 5-methylcytosine first in hydroxymethylcytosine, and further oxidation can then generate formyl and carboxycytosine. Now, there's three enzymes that can do this, TET1, 2, and 3. And important to mention also is that this carboxycytosine can serve as a substrate for a glycosylase, thymine glycosylase, that can excise this base from the DNA, and then base excision repair can then result in the replacement of this base with a non-methylated cytosine. So this TET enzyme coupled with glycosylase activity and base excision repair represents an active DNA demethylation pathway. What was not known is whether this oxidized bases of methylcytosine, whether they have specific readers, right? So we know that methylcytosine is recognized by proteins, but it was not known whether these oxidized states also have specific readers. So we wanted to address this using this quantitative mass spectrometry workflow. So the question we wanted to ask was, which proteins can interact with or are repelled by 5-methylcytosine and its oxidized derivative in differentiated embryonic stem cells? And we decided to focus on ES cells, neuronal precursor cells that can be cultivated from ES cells, and also using adult mouse brain. And the reason why we choose this neuronal pathway is that this is the cell type, so ES cells and neuronal cells is where hydroxymethylation is particularly abundant. And I'm not going to show you all the data, I'll just show you this summarizing slide. This work was published earlier this year. The take-home message is that we see a high degree of dynamics in these interactions. So the readers we identify for methylated DNA in ES cells are quite different from those we see interacting in neuronal precursor cells and in adult mouse brain tissue. And there's another layer of dynamics, because what we also observed is that the readers for methylcytosine are only partially overlapping with those we find interacting with hydroxymethylcytosine. So at least from a biochemical perspective, methylcytosine and hydroxymethylcytosine are behaving quite differently from each other. Now, when we obtained this data, we also started wondering, because if you look at these interactors that we find binding to methylated DNA, they're not all necessarily associated with transcriptional repression, right? We have a number of transcription factors, such as these KLF proteins, RFX proteins, FOXKs, etc., that bind to methylated DNA, but they are known to be transcription factors that can also activate transcription. So we started wondering whether DNA methylation is actually always associated with repression of transcription, or whether in some context it may also serve to activate or at least not repress transcription. And this was also a question that kept a collaborator of ours busy, Osrin Bogdanovic, So he used this transgenic approach. So what he does here is that he takes a reporter construct containing a CMV promoter, and the CMV promoter contains a CpG island, and he then in vitro methylates this reporter construct using a bacterial methyltransferase. And he also uses another construct that is the same plasmid, but now it's not methylated. And on this reporter plasmid is also an acceptor site for a transposase. So then, this is a zebrafish. In zebrafish embryos, he injects these constructs into the developing embryo, and these reporter constructs then get stably integrated into the zebrafish genome. And the promoter is driving GFP and cherry expression. So you can then use immunofluorescence as a readout for the activity of these reporter constructs. And what you can see is that in the early zebrafish embryos, so these are the dome stage as they call it, you can see that it doesn't matter whether this construct is methylated or not. Both of these constructs are equally active in the early stage embryos. Only later during development, this is 24 and 48 hours after fertilization, you can see that the methylated transgene becomes potently repressed, whereas the unmethylated reporter remains transcriptionally active. This is quantified over here. Also important to mention is that in this dome and 48 hours post-fertilization embryos, that the reporter constructs are indeed remaining methylated. So apparently there's a temporal uncoupling between DNA methylation and repression of transcription during zebrafish development. And this is actually also observed in Xenopus embryos. And now we started wondering whether this differential activity may be explained by the fact that different interactors are present in the early stage embryos compared to the later stage embryos, and that determines, so that these readers then determine the transcriptional output of these reporter constructs. So we then wanted to use this quantitative mass spec workflow that we have to try and address this question. So what we did was we took a lot of embryos, so 100,000 of these dome stage embryos and 25,000 embryos 24 hours post-fertilization. We collected these embryos and then generated nuclear extracts from these embryos. And we then subjected these nuclear extracts to DNA affinity pull downs. So we did a triplicate pull down for unmethylated, with an unmethylated DNA construct, then a triplicate with methylated DNA and hydroxymethylated DNA. And then we perform a mass spectrometry analysis of the bound proteins. And because we do triplicates in these experiments, we can use t-test-based statistics to determine which proteins are differentially interacting with either of these three baits. And this is how you can visualize this data. This is a so-called volcano plot, where we plot the ratio of all the proteins we identify. This is in a 24 hours post-fertilization embryo against the p-value of the t-test that is done. This is a so-called volcano plot visualization. And the proteins binding to methylated DNA are in the right of the figure, whereas the proteins that bind to the unmethylated DNA are in the left of the figure. And we can see a lot of interactors, some of which we had also observed in the mouse study. You can see here MBD2, URHRF1, some known methyl CpG binding proteins. One important exception is MBD3. So MBD3 in mouse and humans has a point mutation in the MBD that resulted in a loss of methylation binding, but in zebrafish this point mutation is not occurring, and therefore MBD3 in the zebrafish is a very potent reader of methylated DNA. And in this part of the figure here you can see proteins, a lot of these proteins carry the CXXC motif and also interactors of these proteins. And we can also now visualize this data in this hierarchical clustering. So here you can see proteins binding to methylated DNA in the dome stage embryos. And in this second lane, you can see the proteins we identify in the 24 hours post-fertilization embryos. And in the third lane, you can see the readers we identify for hydroxymethylation in zebrafish. And again, as we have observed in mouse, we see a high degree of dynamics of these interactions. So we have a large cluster of proteins that are dome-specific, and we have a large cluster of proteins that are binding to methylated DNA only in the 24 hours post-fertilization embryos. And we also see dynamics if we compare the readers for methylated compared to hydroxymethylated DNA. There's a large cluster of proteins specific for hydroxymethylation and also for methylated DNA. Now, one of the obvious explanations for these differential interactions is that these proteins are regulated, their expression is regulated during embryogenesis. So we also profiled the abundance of all the proteins that we have here in the nuclear extracts using mass spectrometry. And we can only explain about 50% of these differential interactions by a regulation of these proteins at the protein level, right? So 50% of this dynamics can be explained by the fact that these proteins are present in dome but not present in the 24 hours post-fertilization embryos and vice versa. Now we have this data. We can also compare this data to the interactors we obtained in mouse embryonic stem cells, and we see a large degree of overlap. So about 50% of the proteins we identify in the dome stage embryos, we also identify as binding to methylated DNA in the mouse embryonic stem cell case. And the same is true for these 24 hours post-fertilization embryos. If we compare these interactors to the data we obtained in neuronal progenitors, we see that around 50% of the readers that we identify in zebrafish, we also observe in mouse embryos. And now we have a large number of interactors. We can also look at the Go annotation of these readers to see what kind of processes these readers are associated with. And in pluripotent cells, so this is both the dome stage embryos and the mouse embryonic stem cells, we get Go terms enriched as regulation of transcription and not much else. But if you look at the readers that we identify both in NPCs and also in zebrafish embryos, you can now see Go terms being enriched that are related to differentiation, so embryo development. So it seems as if these readers for methylated DNA that are binding to the methylated DNA only in the differentiated cell type, so the later stage embryos, these readers are actually directly involved in the process of differentiation. And also important to mention, as we have also observed in the mouse situation, is that readers for hydroxymethylated DNA here, in the bottom here, they strongly enrich for proteins that are related to DNA repair, DNA recombination, DNA demethylation. Again, emphasizing that hydroxymethylation is most likely serving as an intermediate for active DNA demethylation. Now, this conservation of the readers is also visualized in this table here that summarizes proteins that we identify both in mouse and zebrafish as binding to methylated or unmethylated DNA. And there were a couple of proteins that struck our attention. That's this, where is it? FOXK1 and FOXK2. There's FOXK2 and somewhere here's FOXK1. So we identify these proteins as constitutive readers for methylated DNA, both in pluripotent cells and in differentiated cells. And these proteins were not previously known to bind to methylated DNA. Now these proteins, FOXK1 and FOXK2, they are part of the fork-head box transcription factor family. These transcription factors are characterized by a fork-head box DNA binding domain, and this domain is also known as a winged helix. Now this was interesting because we previously showed that a group of proteins, RFX proteins, they bind with a high affinity to methylated DNA using this winged helix domain. This is an NMR-based structure determination of the RFX5 DNA binding domain with binding to methylated DNA here indicated in green. So we started wondering whether these FOXK proteins may actually be direct readers of methylated DNA. So we addressed this both in vivo and in vitro. So this is just data, uncode data, so publicly available data that we mined. So a FOXK1 ChIP-seq that has been performed in HeLa cells. And what we see is this very interesting bimodal distribution. So around 50% of the genome-wide FOXK1 binding sites are characterized by low levels of DNA methylation, and another 50% are characterized by high levels of DNA methylation. We also express this FOXK1 fork-head box recombinantly in E. coli, and we could validate that indeed this winged helix displays potent methylcytosine binding specificity. And this is also uncode data with methylomes generated in different cell types. And you can see this bimodal distribution. So around 50% of the FOXK1 binding sites have high levels of DNA methylation and 50% have low levels of DNA methylation. So we were interested in these proteins to study whether they have an important function during development of the zebrafish embryos. In zebrafish, there's 3 FOXK proteins, FOXK1, FOXK2, and FOXK2-1. And if you knock down these proteins using morpholino separately, you don't get a phenotype, so there's probably some redundancy between these proteins. But if you knock all 3 proteins down at once, you get a very strong developmental phenotype. So these zebrafish don't reach maturity. Now what's interesting is that we can rescue this phenotype by expressing in the zebrafish embryos a human a cDNA encoding FOXK1. And this rescue is dependent on DNA binding, because if we try to rescue this phenotype with a FOXK1 protein that contains a point mutant that disrupts DNA binding, we can't rescue the phenotype. Now, we also did an RNA-seq analysis of this knockdown phenotype at an earlier stage than you see here. And there's a cluster of proteins going up and a cluster of proteins that go down. And if you look at the gene ontology of these cluster of proteins that are downregulated upon FOXK knockdown, you enrich for terms related to cell division and mitosis. So apparently, in the developing zebrafish embryos, these FOXK proteins are regulating genes that are important for normal cell cycle progression. Now, the final question, of course, that we wanted to ask now is that can we now use this information that we obtained in the quantitative, using this mass spectrometry, to manipulate this transgenic system that I showed you in the beginning? In other words, can we now repress the methylated transgene by overexpressing 24 hours post-fertilization specific readers in these early dome stage embryos? And the prime candidate for this experiment was MBD2, because MBD2 is a known methyl CpG binding protein, known to assemble in the NERD complex that represses transcription. And we only identify MBD2 NERD binding to methylated DNA in later developmental stages, so in the NPCs and also in these 24 hours post-fertilization embryos, but not in pluripotent cells. So this is the result. It's pretty clear. So these are these dome stage embryos again. Here you can see expression of the methylated transgene in these dome embryos. So it's nicely being expressed. But if we now, in this dome stage embryos, also overexpress MBD2, we can completely silence the transgene now in this case. And this is a control here showing that overexpression of MBD2 does not affect expression of the non-methylated transgene. So overexpression of MBD2 can induce 5-methylcytosine-dependent repression in the early embryos. That all brings me to some conclusions. So I've showed you that during zebrafish development, there is a temporal uncoupling between DNA methylation and repression of transcription. An important message is that these readers for methylated DNA are highly dynamic during zebrafish development, which is very similar to our observations we made in the mouse. I showed you that we have two novel methyl CpG binding proteins, FOXK1 and FOXK2. Depletion of these proteins impairs normal zebrafish development. These proteins regulate genes related to cell division. And one conclusion that at least I draw from this work is that it appears that the functional readout of DNA methylation is heavily affected by the repertoire and abundance of different readers that are expressed at any given time in a cell or a developing organism. I have to thank the people who did the work. So the first part of the work, the mouse study, was done by a very talented graduate student, Nelleke, and the zebrafish work in my group was done by another graduate student, Arne Smits. And we have very nice ongoing collaboration with Thomas Carell, who synthesizes these methylated and hydroxymethylated oligonucleotides for us in Munich. And this was, the zebrafish study was a very nice collaboration between our lab and Osrin Bogdanovic, who was in the Scameta lab in Seville. This is Osrin at Miami Beach, having a good time. And these organizations are thanked for funding, and thank you for your attention.
Right.
Regarding the FOXK1 data, it wasn't clear to me, did you show that it binds methylation dependent, or is it just not caring if it's methylated or not?
So, yeah, that's, the slide is gone already. So it's bimodal. So 50% of the binding sites, genome-wide, are hypomethylated, and the other half is not. Now, currently we're trying to look whether we can sort of look for motives in the cluster. Yeah.
Could it be not just insensitive to methylation?
Sorry?
Could it not just be a transcription factor that simply is insensitive to DNA methylation?
That could also be an interpretation. I mean, what we clearly see is that transcriptional activators that are not have an affinity for methylated DNA, like TAF1 in this example here, there you see a strong correlation between hypomethylated sites, right? Then you have MBD2, which is exclusively bound to methylated sites. And then this protein shows a bimodal distribution. Now, whether these 50% of sites that contain DNA methylation, whether the methylation of those sites is important for FOXK1 to be there, that we don't know.
So how prevalent do you think this mechanism of essentially changing the output of an epigenetic modification? Is this something that's occurring early in embryogenesis, or is it something that you think is occurring, say, at the terminal differentiation stage?
Yeah, I mean, one thing talking about epigenetic modifications in general, this is something that is very specific for DNA methylation. So we also study interactions with methylated histone tails, for example. And there, if you do the same experiment with pluripotent and differentiated cell types, that the readers we find in that situation are mostly overlapping. So this is something that is quite specific for DNA methylation, this high degree of dynamics. And so, yeah, this is what I can say.
So you don't think that histone modifications are being, the functional consequence of them are being mediated by differential expression of those readers? Like, for example, polycomb group. Polycomb group seems to have a differential effect in ES cells than it does in terminally differentiated cells in terms of its ability to repress gene.
There's a subtle subunit composition change of polycomb during differentiations, right? So PSC1 contains CBX7 in pluripotent cells and CBX4 and 8 in differentiated cells, and that may, fine-tune the localization of polycomb complexes during differentiation. But it's a subtle change. I mean, this broad, very minor overlap in readers in early and late stage is very specific for DNA methylation. So, you may interpret that how you want, but I see that DNA methylation and the dynamics thereof are the main drivers of differentiation rather than differential interactions with histone modification.
Okay. There's one.
Are there DNA motifs which predict the binding of certain of these readers so that one can make a judgment whether a certain, for example, polymethylated region is an active and not a repressed site?
So that's a very good question. So I should mention that all the data that I showed you here is basically using a single bait with 4 CPGs in a short stretch. There's going to be readers that are caring about the surrounding sequence of these CPGs, whereas other proteins, like the classical MBD proteins, they're binding all the time to these methylated CPGs, regardless of the surrounding context. So at the moment, we're extending these studies by using multiple baits, mapping to genome loci that carry differential methylation during differentiation, and doing many of these baits, and then trying to see which readers are always there and which are context specific. So that's what we're doing at the moment.
All right. I would like to thank the speakers again for an excellent session, and